Dear colleagues, dear audience, let me start by saying how deeply honored I feel to be with so many talented people, young ones and even younger ones. When I'm asked what we do in the laboratory, what we study, then it's a difficult question to answer. When I shed my shyness, then I just say that we study the most important questions of life and death. And that is not really far from the truth, because, but it's true only at the level of the cells, to be honest. Because we study mechanisms that make the decision between life and death, and also between the different forms of cell death, because it very often does make a difference. It is important how cells die. If a cell dies by so-called apoptotic cell death process, it often remains almost unnoticed in a tissue or in an organ, because these cells are removed very efficiently from the organs, whereas a necrotic cell death, when the cell falls apart and the content is released, that causes secondary inflammation and secondary damage. So that's something that we should avoid at all costs. So we study this in the context of DNA damage. Oops, sorry. I think I just went backwards. We study all this in the context of DNA damage because our cells are constantly exposed to various DNA damaging stimuli. It can be ionizing radiation, reactive oxygen species, UV radiation, different chemicals, mitotic stress, and the likes. And this triggers the DNA damage response, which may have two outcomes. Either the cell is repaired and survives, or if the damage is too severe, then death must be the consequence. So we studied this in the context of oxidative stress, when reactive oxygen species or reactive nitrogen species are overproduced, causing DNA breaks, and the activation of our favorite enzyme called poly-ADP ribose polymerase 1, which catalyzes poly-ADP ribosylation. Well, it sounds terrible, but it's not that complicated. So that is the pathway that we study in most of the biological models that we use. That's just the chemical part. These enzymes, poly-ADP ribose polymerases, use NAD, one of the key energy metabolites of the cells, and cleave this N-glycosidic bond. So they cleave off nicotinamide, and the remaining ADP ribose units are covalently attached to acceptor proteins, such as PARP1 itself and other nearby proteins. And then further ADP ribose units are added, even branches are introduced. So a nice branched poly-ADP ribose polymer is formed in response to DNA damage. Many years ago, we created an animation that shows the key events in this pathway. So I would just like to share with you this animation. Right now, it's just reactive oxygen species, superoxide, hydrogen peroxide are coming out of the mitochondria, but it can also come from external sources. Some of the species can reach the nucleus where they cause DNA damage. And this is where our favorite protein comes into the picture. It recognizes the DNA breaks and becomes activated. Once activated, they start using NAD, and from NAD they create poly-ADP ribose polymer attached to themselves, nearby proteins, histones, and this way it marks the site of DNA damage so that real effector proteins that can repair come, they find a place, they come here and repair the damaged site. We can study this and the kinetics of this pathway using a technique called microirradiation. My colleague Mate Demein set up this model in our laboratory. So here he just uses the laser from the confocal microscope and can cause damage in a small area. This is just one cell nucleus that we see here, and it's a little bit red because our favorite enzyme carries the red fluorescent tag in this cell. So when we apply damage to the nucleus, you can see that as early as 4 seconds after the damage was done, the PARP already appears in the spot, and then it just keeps accumulating in the site. And when Mate is in the playful mood, then he creates this damage in 2 spots and a curved line, so he can make a smiley on a cell nucleus. So this is just a single nucleus that we see here. Okay, so that is the background. So what's the function of this process? Of course, one function is what we have seen. It's the DNA repair pathway. It does not directly repair the damage, but it contributes to it by DNA sensing. But it also has lots of other so-called moonlighting jobs, such as the loosening of the chromatin, gene regulation, protein stability. Lots of other functions can be linked to this protein. I don't have time to talk about those because I would like to share with you some data on, wow, that looks terrible. Let's see what comes out of that. So what keeps me fascinated about this protein is that on the one hand, it's a cell survival protein through the DNA repair function. On the other hand, it can kill the cells in a very bad manner using the necrotic pathway. And this duality is what fascinated me in the last couple of years, to put it this way. It's a DNA repair mechanism, so people very early on suggested that inhibition of PARP1 might be a good idea if we combine it with DNA-damaging chemotherapeutic agents or ionizing radiation. This way we can sensitize the cancer cells to death. And this idea worked quite well. It was not like a breakthrough, but it was something that many groups have demonstrated. However, the real breakthrough came when Thomas Halliday from Sweden has shown that we should take a more targeted approach to this question. And let's just try PARP inhibitor therapy in a set of tumors that carry mutation in the so-called BRCA gene. The protein product of this gene is involved in a DNA repair pathway. Let's just call it homologous recombination pathway. And our favorite enzyme is involved in the base excision repair pathway. So if the tumor cells carry the mutation and we inhibit the alternative pathway with a PARP inhibitor, it should lead to cancer cell death. And this concept worked, and this paved the way for the introduction of a new novel therapy for cancer, so that today we already have 4 PARP inhibitors available for the therapy of such tumors. That was a real breakthrough in the therapy of BRCA-deficient ovarian and breast cancers especially. So we didn't do too much cancer-related work before, but recently we had a project that focused on the use of NAD, which is the substrate of the PARP enzymes. And the idea was to target PARP activation by limiting the supply of NAD. There are several enzymes involved in the NAD synthesis, so we focused on NMN81, because it's difficult to bypass in the synthetic pathway. And we just knocked out the gene of the nuclear isoform. NMN81 is the nuclear isoform, so this way we thought that NAD can be depleted from the nucleus, and this way PARP1 will be indirectly inhibited, and that might have similar consequences as a direct PARP inhibition. So what my colleagues did in the lab, they used this CRISPR-Cas9 technology. They knocked out the NMN81 gene. So we had a parent cell line, an osteosarcoma cell, and one that did not carry the NMN81 protein. And then we just compared the sensitivity of the cells to a chemotherapeutic agent, cisplatin, and the cells were indeed more sensitive than the parent cell line. Interestingly, the NAD content was smaller, but there was not much change in response to cisplatin. There was a big change in ATP. We still don't understand how it works, but we just found it interesting. But what about DNA damage in these cells? Look for the green spots here. The green spots represent DNA damage. In the top line, the parent cell line shows some DNA damage, but in the NMN81 knockout cell lines, you see much more damage, and this is the quantification showing the big difference. So is this because the PARP activity is limited? Yes, it might be, because in the parent cell line, this therapy caused lots of polymer synthesis, but there was hardly any in the knockout cell line. And if we just quantify cell death, there was some in the parent cell line, but much more in the NMN81 deficient cells. So this might be a new approach. Well, I'm afraid I'm not going to be able to show you this. So I will just skip that. Wow, never happened to me. But with this image, I can just illustrate the fundamental effect of PARP1 on cell death. In a severe oxidative stress situation, it causes necrotic cell death. So these are electron-microscopic images of thymocyte, a model cell line, a cell type. Very severe necrotic phenotype. When we focus on the mitochondria, they look terrible. They just swollen. The swollen mitochondria lost the cristae, so all the ultrastructure is destroyed. If we do the same intervention with thymocytes that do not have a PARP1 enzyme, knockout animals, then there was hardly any damage. So that was quite a striking thing, an experiment I did when I was much younger than now, and it still keeps me fascinating. It's because this is not just something that happens in a model cell line. You see the very same thing in the myocardium. This is myocardial injury. This dark area is all necrotic area. If you inhibit the enzyme with a compound, much less damage can be seen. If you look at a stroke model, these are sections from the brain. You can cause the necrotic death of a big area by occluding an artery in the brain, and you can save much of this if you apply a PARP inhibitor. Or inspired by Professor Roska's excellent lecture, I also included sections from the retina, from a retinal cell death model, showing PARP1 activation and cell death, and I think it's quite obvious that these two occur in the same cell line. So this is something that can be observed in many different models. A couple of years ago, Professor Hegyi got me involved in a project, so I had to do some pancreatitis research, even though I'm not an expert in that, but I just viewed this as a model for necrotic cell death, because in acute pancreatitis, many cells die, they release the content so that it appears in the circulation. So there's lots of necrotic cell death going on here. Looking at the role of PARP1 in this model did not make much sense because a Spanish group in Barcelona, led by my friend Jose Yalamos, already showed that PARP activation contributes to inflammation and tissue injury in acute pancreatitis. So instead, we just focused on proximal events such as reactive oxygen species production. And interestingly, we found many compounds that are known to scavenge, to capture free radicals, but did nothing in this model. But one of them, this flavone compound, tristatin, managed to reduce inflammation. Interestingly, it also had a direct inhibitory effect on the PARP enzyme. I don't show you details on this project, but I'd like to show you some details on this one, where we looked at this enzyme, which can recognize oxidized bases in the DNA, oxidized guanine, and can cleave them off, remove them. In turn comes an endonuclease that creates cleavage, and this is actually what activates PARP and not the direct damage of the damaging effect of the free radicals. So we just used an inhibitor of this oxoguanine glycosylase enzyme and caused pancreatitis using the model that we learned from Professor Hegyi and Professor Mallet. So we saw lots of damage, lipase release from the pancreas, amylase release, and infiltration of neutrophil granulocytes. It was all there, and the compound quite efficiently reduced this damage signs and inflammatory signs. So if we looked at the histology, we could see the severe damage, and there was much less damage when OGG1 was inhibited. Also, this inflammation and damage markers could be quantitated and showed the same thing. What about poly-ADP ribosylation? Well, indeed, there was lots of poly-ADP ribosylation in the pancreas, much less when OGG1 was inhibited because the oxidative guanine remained in place and no endonuclease could cleave the DNA and activate PARP1. And finally, let me show you some data emphasizing the importance of PARP1 expression. PARP1 is a protein that can be found everywhere. At least at the medium level, you can find it in every single organ. Some contain quite a lot. So people think that it's an abundant enzyme, so it's regulated at the level of activity and not gene expression. But the thing is that it's not necessarily always the case. For example, we performed experiments with cancer cells that we arrested in the G1 phase of the cell cycle. So that's the interphase that separates two mitotic phases, and we can stop the cell cycle in this phase by using inhibitors of cyclin-dependent kinase 4 or 6. So these were the substances that we used to stop cells in this G1 phase, and we wondered how their cell death sensitivity changes. So we applied various various genotoxic stimuli, and we found that all of them, so when we stopped the cells in G1, they always became much more sensitive to all of these therapies. Why is that? It's because in this G1 arrested cell, the expression level of PARP1 is much lower, so it's expressed at a lower level, and this might be the cause why they were more sensitive. So we wondered what happens on the promoter region of this gene. Why is it downregulated? So we looked at several components and epigenetic marks to see what's going on in this promoter. And just to cut this story short, I just show you the scheme that we came up with at the end. So with this chromatin immunoprecipitation experiment, we managed to identify the marks and the proteins that are present on the PARP1 promoter that switch it off, and what the composition of this complex is when the PARP1 promoter is on. So that was a rather new finding showing that PARP1 is regulated at the level of gene expression as well. Recently, we do lots of work on macrophages, phagocytic cells that are master conductors of the immune response, especially the innate immune response. The precursors are called monocytes in the circulation, and when they leave the circulation, in the tissues they differentiate to macrophages. And depending on the environment, they can take various different phenotypes, like the inflammatory one and many different anti-inflammatory phenotypes. So we thought that the pathway that we study might be involved in the defense of the macrophages from the oxidative stress. Because these M1 inflammatory macrophages must work in an inflammatory environment, so they must be resistant to oxidative stress. And indeed they are. So these are just regular macrophages. The red color indicates cell death, and they can be killed, as you can see. But if we send them towards the M1 phenotype, hardly any cell dies. You can see the striking difference here between resistant and sensitive cells. So how do macrophages become resistant to this cell death? It's because they managed to downregulate PARP1, so there is no poly-ADP ribosylation, unlike in the M0 macrophage phenotype. So again, PARP1 downregulation can make macrophages resistant to cell death. So finally, I would just like to acknowledge the excellent work of colleagues who work in the lab. Some of them already left, so this is not the latest image, but they do the excellent work. And I'm really thrilled to work with so many talented National Science Academy students whom I supervise, co-supervise, or simply work together. It's a really excellent bunch of people. And finally, I'd like to acknowledge the financial support of the various organizations. Thank you very much.
Thank you very much, Professor Virág, for this nice presentation. We have time for a couple of short questions. If you have any, please raise your hand and indicate that you, please.
László, I think I can start with this classical introduction. Before your talk, I was confused. After listening to your excellent talk, no, I am still confused, but a much higher level. So just give me a response to a simple question. With PARP inhibitors, there are drugs for the treatment of cancer. You have shown at least 4 of those. And I wonder about their stages, where they are now in the clinical practice. If I understood you correctly, they have been considered in breast cancer.
They are used in breast cancer and ovarian cancer. So they are on the market. These are the clinically available ones.
Mm-hmm. But what makes me confused a little is that you say that they sometimes implicated in cell necrosis. How can it be directed towards cancer cells, and what is the collateral damage? Or so, how should we interpret this factor in their activities?
Yes, I think it's important to emphasize that this pro-necrosis effect of PARP activation depends on the severity of DNA damage. With chemotherapeutic agents, or even with ionizing radiation, we are not even close to the level where necrosis could be caused. So these agents typically cause apoptosis or some similar more or less harmless cell death pathways, which can be enhanced by PARP inhibitors. In this case, we don't need to consider necrosis as a potential outcome. It's apoptosis or more apoptosis if PARP1 is inhibited. It's a different cup of tea when you have a severe oxidative stress situation, myocardial ischemia, reperfusion injury, stroke, or other models, Parkinson's, degenerative neurological diseases, where cells die in a necrosis-like way. It's called partenitis. I could talk a lot about it, but I don't have time. So that's a different cup of tea when severe oxidative stress causes necrosis. You don't need to consider that in a cancer, even if it's treated with chemotherapeutic agents.
Thank you very much. Now I'm not confused at all.
Thank you very much. We used up our time, but if you have any question for Professor Virág, you can find him in the coffee break. And now, on behalf of the National Scientist Academy, we would like to, don't go away, we would like to recognize your excellent contribution on this conference and in general in the program with a diploma.